with the very famous Supratman. So hopefully we're going to do some interesting research on Aglaya. So I think everybody knows Aglaya is very interesting for cancer. So it could be very interesting. And then I'm going to have a new postdoc starting in 2025 for fungal natural products. So I don't know, tidak tahu belum. Mungkin mungkin tiga hari, tiga tahun. Okay. So okay. Yeah. So the question for this lecture is we're going to be talking about fungicides in Indonesia. Yeah. So we want to maybe do some research on the potential of plants to be fungicides in Indonesia. So first of all, we have to say what is the problem with fungicides in Indonesia. Well, I can give you 3 examples. Nitroglycerin is used in Indonesia, but many of them Pakistan also. So there's a problem with this one. Chlorothalonil is very popular fungicide in Indonesia. But I don't know if I can. Yeah. Propionate is also very popular in Indonesia. So that was farming agriculture in Indonesia dan ini banyak ya. Okay. And then another problem. Ada masalah di triazole fungicide. Kamu tahu triazole? Ya. Okay, pertanyaan apa masalah dengan triazole? Bukan kan? Ya. Jadi, okay. So what triazole is in agriculture. We're using this triazole similar. And in rumah sakit, also triazole for fungal infection in humans. Yeah. So if you use this triazole in agriculture, the problem is that fungal infections for humans become antibiotic resistant. How do you say this? How do you explain this? So basically, we're using this similar compound in agriculture and similar compounds in hospitals. So then we have natural selection and fungal pathogens become resistant. So basically, aspergillosis is a fungal species in agriculture, but also in humans. So the problem is that basically, if we keep on using triazole, then we can't use triazole for agriculture, and it's not workable. It's not So the big problem is let's see if I can translate this online. How we can say that the emphasis, the compound pathogen in a hospital is adapting to learning how to, it's adapting to the triatal, becoming resistant. So becoming basal resistant. Okay. So this is one big problem. So what is the potential solution to this problem? So the problem is this one. Maybe the solution is tidak yang dari tulang dari alternatif yang popular ya. So here are some examples. One to do an example of natural fungicides. So you can see these products are already available on the market in Europe and in North America. So what compound is present? So this one we see transneuronal. Yeah, this one here is terpium for monoterpene, and this one fracture is a peptide. Yeah, so this is the existing example in the world of a natural botanical fungicide. So one question, pertanyaan, okay, dari Indonesia ada banyak pohon ya. Jadi mengapa tidak ada fungicide natural dari Indonesia? Belum. Okay. So lihat ini transneuronal terpium for pertanian dua senyawa bekerja pada fosfolipid bilayer ya. Kamu semua tahu fosfolipid bilayer? Ya. Jadi dengan tidak kerja. Okay, jangan khawatir. No. It's okay. Yeah, it's just the pointer doesn't work. Yeah. Okay. Jangan khawatir. So these compounds are disrupting the phospholipid bilayer, and then the fungus is dying. Yeah, so when the phospholipid bilayer is broken, the fungus dead. Yeah. So how about if we want to ask the question, how can we know if the phospholipid bilayer is broken? And how do we say phospholipid bilayer is broken in Indonesia?
Jadi merusak lapisan fosfolipid dan dua lapisan fosfolipid.
Yeah. Okay. So we can do this. We can use propidium iodide. Okay, so misalnya propidium iodide tidak bisa. Thank you. Okay, so any propidium iodide. Okay. Yeah, any propidium iodide So propydium iodide, kamu tahu propydium iodide? Bukan? Okay, so think propydium iodide, kapan propydium iodide masuk di sel, propydium iodide fluorescent, ya. Tetapi kapan propydium iodide bukan masuk di sel, bukan fluorescent. Ya, jadi kalau apa ini phospholipid bilayer sudah apa? Rusak, okay? Propydium iodide bisa di sini propydium iodide kerja fluorescent, ya. So lihat, ini saya kerja ini sudah dengan SDS. Okay, kamu tahu SDS? Sodium dodecyl-sulfate. Yeah. So sodium dodecyl-sulfate is a membrane disruptor, phospholipid bilayer disruptor. So saya sudah membuat ini. Okay, ini untuk 20 menit, 50 menit. Okay, ini control. Okay, ini fluorescence, ya. Dengan plate reader. The phospholipid bilayer. But now, yeah. Okay. So, wait. So any example dan any example can just be phospholipid bilayer. Okay. Tetapi any example, okay, bukan phospholipid bilayer. Any peptide can just be how do you say iron? Same. Okay, sama iron. So any peptide kerja the iron chelator. That means like how would you say iron chelator? It means it takes the iron. Yeah. Okay. Jadi okay. So saya kerja satu publication dengan ini fungicide nama fracture. Ya. Jadi kami sudah membuat transcriptomics. Okay. Karena tahu ini produk ini fungicide fracture tidak tahu berapa kerja, ya. Jadi kami sudah membuat transcriptomics dan kami sudah lihat ada banyak genes functional to iron, ya. Jadi kami pikir mungkin ini fracture kerja di iron chelator, ya. So kami sudah membuat eksperimen dengan supplementation with iron, and this sudah tidak kerja dengan ada banyak iron. So kami sudah pikir fracture ini peptide kerja di iron saja. Oke, so bertanya, bisakah kita menemukan fungisida nabati dari tanaman Indonesia untuk melindungi tanaman cabai? Cabai, cabai. Maafkan saya. Ya, ya. Oke. So di sini mungkin ini this is our project proposal. Yeah. So step one, we want to collect 5,000 Indonesian plants from Kebun Raya Botanic Garden. Yeah, Kebun Raya Bogor. Okay, 5,000 besar sedikit ya. Tetapi mungkin dengan impact student, mungkin satu student bisa membuat collection mungkin 50 sampel setiap hari. Okay, mungkin impact student, mungkin dengan impact bulan kami bisa membuat 5,000 ya, 5 juta ya. 5,000 ya. Okay, and then extract the sample and we put them into 96 well plates. So this we call like an extract library, and then screening the sample with fungal pathogen. So I think Dr. Ender can do this experiment. Isolation of the active compounds, Professor Unai, with some students. And then more testing with fungal pathogens. And then at Q, we can do toxicology of the active compound. And then in UNPAD, we can do testing on plants. And then in we can also do mechanism of action, which is important to understand the mechanism of action. Yeah, so 4 students, 25 samples, 3 bulan, mungkin ya kami bisa kerja ini.
Recording stopped.
simple protocol, but we can get results in plants. Okay. So now we have from 5,000 plants, we have active compounds that works in plant infection with fungus. Oh, I can capture this. Oh, okay. So another question is the toxic? So I have a collaborator, we can do predictive toxicology. So a new idea for toxicology, this is like a new idea is we can make a prediction using artificial intelligence. So the computer can understand, based on the structure, does this compound look similar to another compound that is a problem? So the computer can make some kind of prediction about toxicity. Yeah, actually, one more thing I would say about this. So this is like a big idea at the moment is in silico pharmacology. But the only problem is it's based a lot on synthetic compounds. So if you have a natural compound, maybe the computer doesn't understand because it can't find a similar compound. So if you're testing synthetic compounds, the computer can make a comparison with other compounds. If you have a novel plant compound from Indonesia, maybe the computer does not understand what this compound is. That's the only problem, I think. Uh-oh. Yeah. Okay. So last question is we want to know, how does the fungicide work? How does the chemical kill fungi? Why do we want to know this? Because your fungicide might be accepted. Probably the government wants to know how does this fungicide work at the molecular level. And if they want to have a good application, it's important as well. So this is what one of my areas of interest. So I think this is kind of work a little bit complicated to explain what we do with this compound, and we give it to